So right now I want to show you how to use the library to process and import your experimental data. So let's imagine you have an experiment that you just did. You want to analyze it. So first thing you want to do is to create a new experiment, which you can do by using this function called initialize experiment. So you can create a new script or write it down directly in the command window. So the xpd, which is the argument of this function, is actually the name you want to give to this experiment. In my case, I'll choose February 2021, and you can just run it. So once the experiment has been initialized, you can go to your data path, and you'll see that a new folder has been created for the experiment, with inside some other folders and files. So this is the folder of your new experiment. You have a process folder, which is where the library saves its results, its process data, like receptive fields or spike trigger averages or spike times of your cells and so on. The sorted folder instead is where the program looks by default for the raw data or the sorted data, meaning the results of the spike sorting. Now that your experiment is initialized, next step would be to tell the library which stimuli you actually displayed in this experiment. You can do that by filling this steams.txt file, which is a configuration file where you can specify which stimulus you played and which stimuli you played and in which order. You can follow the instructions here in the comments. Basically, each line corresponds to a section of the experiment, and you just write down the name of the stimulus that you played. So for the experiment I want to analyze, it would be checkerboard first, then the full field, then 3 sets of flashes, and then the full field was played again. Now not only you can specify which stimuli you played, but also under which conditions. So for example, in my case, the second set of flashes was played after the application of pharmacology, and the third one was played after the application of a second type of pharmacology as well. Same thing goes for the last one. And you can also add comments like this. So once you have saved all of these and make sure it's correct, you can close it, and you can initialize your sections, meaning basically the library will read your steams file, so it will understand which stimuli you played and in which order, and it will initialize the sections of your experiment. So I can do initialize sections xpd. So like this. I receive a message: the section for the experiment has been initialized. So I can go back to my experiment, and you'll see that inside processed you have a different folder for each section. So checkerboard, Euler, 3 flickers, and another Euler. So now later on, when we will process the actual data, we'll see that the code will save in each of the section some particular information. In particular, if your stimuli have some repeated portions, you can compute the which are the timestamps of all the repetitions of your stimuli, so that then you can generate PSTHs and raster plots. Now all of this, we will see it later on, is handled automatically by the library. So if you go into your project folder, you'll see there's a folder called stimuli. Inside here you have all the stimuli that are known by the program. So right now it's just the checkerboard, Euler, and the flashes. And inside each of these folders there's some information about the stimuli. In particular, there's a mat file that tells you the structure of the repetitions, meaning that it will tell you, given the event marker of the stimulus, where to look for the repetitions, so that you can then extract them and generate the PSTHs and raster plots. So it's not important right now to go into the details of this. It's just important to know that if you use that the program will just read your stims file, it will see if there is some stimulation that matches with the ones that he knows, like the checkerboard and the Euler, and if it's the case, it will automatically compute the repetitions, and so you will be able then to plot the rasters and the PSTHs. If you play a stimulus that is not known by the library, meaning you don't have the repetition map saved in here, so you will not be able to plot the repetitions or to plot the raster plots, but you can always in a second time add new stimuli to your list of known stimuli by basically just creating a folder and putting inside the repetition mat, which is a MATLAB file. We will see this how to do this later on in another video. So for now it's not important, you just have to know that for known stimuli you will be able to generate PSTHs. For stimuli that are not known, you will have to do one extra step if you're interested in plotting that information. So now we are happy with this, we can close everything, and we can now actually start to import our data. So in order to import your data, you can use this function called extract experiment. This function will extract the triggers from the raw experiment file. It will import the results of spike sorting, and it will compute the repetitions for the repeated parts of the stimuli which are known. Now by default, raw files and results of spike sorting are expected to be found in the sorted folder of your experiment. So here. So you can rather save them, copy them here, or if you want, you can also just use some optional parameters of the function to tell MATLAB in which location you have saved these raw files and spike sorting files, which is what I'm going to do now. So I will do this. So now, not only now I have to give the name of the experiment, but also which one the recording rate of the multilateral array, which in my case is 20,000. And I will also add the optional parameters to tell the library where the raw file is. Now in general, in MATLAB, you can just select a function and click and do Ctrl D to open the source code of the function, and this will basically tell you which are the parameters that you are supposed to use, if there are some optional parameters, and basically it allows you to read the whole documentation of a function. So here I can see what's the name of the optional parameters to find the raw file and spike sorting file, and I can use those. So I'll do raw path. So the raw path for my experiment is in here. Yeah. So here is where my raw file is saved. Here is the name of the raw file, which is this. And same thing for the sorting. So sorting path. So this is the files which are the output files of spiking circus after spike sorting. Same path, same name. And then one last thing, one last parameter that I need to specify is the suffix. So What is this? You might know when you save on spiking circles your results, you can save multiple times in the end, and each time you save basically will create a new version of your results, which, as you can see, they differ by this suffix in the end, which can be -2, -1, or so on. So basically you might want to tell the library which of these suffix is the correct one that you want to read. So in my case the last one is -2, so this is the one I want to use. So this is why I'm specifying here -2. So now I can add these parameters, so row path, Okay, so now I'm ready to extract my experiment. So as you see, it's going. The first thing the program is doing is extracting the event markers for the stimuli. Now these might take a few minutes depending on how big is your raw file and on your computer and where the data is saved. So I'll pause and resume when it's done. So this is the second part of the video to extract data and process the data and the raw data from your experiment. So I just finished extracting my triggers from the experiment, which means basically which are the event marker of the timestamps at which each frame of a given stimulus happen. So once the trigger are extracted, the code groups these triggers into sections. So here we found 6 sections, which means 6 different types of stimuli were played. You can have a look here at the frame rate of each of them. So for example, the first one is the 30, then 40, and then we have the 3 stimuli at 1 Hz, which corresponds to my flashes. So basically, once this is done, the library assigns each of these sections to the labels that we gave before. So it assigns basically to the checkerboard its own event markers, to the full field, and so on. And then it can also, as I told you before, use these event markers to compute the repetitions, the repeated portion of the stimuli, so that we can then be able to plot PSTHs. So once the repetitions are saved, then also the responses are extracted from the results of the spike sorting. So you see all the traces are extracted. In my case are around 400, and then we are ready to go. The last thing that this function does, it also plots some test panels in which you plot just the first 10 cells of your dataset, and it shows you the raster plot and PSTH to the Euler stimulus and checkerboard. So this is just a quick test to make sure your cells, your responses looks as expected. So yeah. So now the last thing that I want to show you, once you have all of your experiment data extracted and saved in your folder, you can also plot PSTHs. So you can, to plot PSTHs, you can use this function shown here, plot section PSTH, like this. So yeah, so the first argument is again the experiment. The second argument is the section for which you want to plot the PSTH. So I already showed you the checker button flicker PSTHs. Now I'm gonna check also the flicker one, and there